Hello, everyone. Okay, so we're going to start our review class for today. My name's Rob. This is Catherine. We're both your TAs for this course. Not all of you guys have us. We're all divided into sections. So I guess we'll get started. So as you know, the exam's on Sunday, October 14th, this Sunday at 1 o'clock. It's a 2-hour exam, and it's located in the Israeli Theater, either room 101, 102, or 301, depending on whether you're in the neuro section, the psych section, and then by your last name, depending on whether you're on the TV or in class. So any questions on that? So what's on the exam? So the exam's 20 multiple choice questions worth 1 mark each. There's going to be 10 definitions, 2 marks each, 6 short answer questions, and 3 figure labeling. So multiple choice is pretty straightforward. The only thing I'm going to say about the definitions and short answer is try to put as much information as you can to get the entire mark. So even though, for instance, each definition is only worth 1 mark, try not to put just like 1 word.
2 marks.
Definition, 2 marks. So make sure you have enough info. On the other hand, try not to put too much, especially if you don't know what you're talking about, because what ends up happening is you might have the first part that you're writing is actually correct, and then if you keep filling it with information that's not right, we won't be able to give you full marks. As for what's on the exam, chapters 1 to 5, and everything from the first day of class till today. So for today, we're going to cover a little bit on gene expression. You guys actually skipped this in class, so I'm going to take it kind of slowly, but it's really only 6 slides, so it's really not that much information. Then we'll go over action potentials again, and then neurotransmission, and then we'll open it up to you guys if you have any questions. Okay, so for gene expression, so when a neurotransmitter binds to a G protein receptor, can anyone tell me what happens after that? Nope? Okay. So what happens is once a neurotransmitter binds to a G protein receptor, the alpha subunit detaches itself from that, and then one of 2 things can happen. So the alpha subunit could bind to a close by ion channel, opening up that ion channel, or it could activate an enzyme, which then activates a series of transcription factors, and these transcription factors then activate certain genes. So before I go into the actual gene expression, I'll just go over the internal cell structure of a neuron. So to use the example from the textbook, you could think of the cell body as a factory, a shipping and packaging factory. So basically you have, I'm going to draw because I like drawing. So you have your nucleus here, which is the central executive. So it contains all the information that the cell needs to function properly. On the outside of the nucleus, I'm going to erase that. Maybe I shouldn't draw because it's taking long. So on the outside of the nucleus, you have what is called the endoplasmic reticulum. So that's where your proteins are actually formed. And on the outside of that, so right here, is a Golgi body, and that's where those proteins are packaged and then later transported to the microtubules, which are here. And then they're shipped off down the axon to the axon terminal. Okay. So again, within the nucleus, it's the central executive. It contains all the information for the cell. And that information is contained on your chromosomes. So we have 23 pairs of chromosomes in every cell. The chromosomes are made up of DNA, deoxyribonucleic acid, which is made up of 4 types of nucleotides. So you have adenine, which is A, thymine, T, guanine, and then cytosine. So adenine always binds with thymine, and G always binds with C. So those are the only combinations that are possible. And then every, am I going too fast? A little bit? Okay. So I'll do that part again. So you have 4 nucleotide bases, A, T, G, and C. A always binds to T, C always binds to G. And then every 3 nucleotides, so in combination, so if you say, let's say you have, let's say an eraser. It's not working. So let's say you have 3 nucleotides. Let's say they're all A's. That nucleotide makes up what is called a codon. And then that codon codes for a certain protein. So a gene is a specific sequence of DNA. So it's a specific combination of nucleotide bases, and depending on that specific combination of bases, it'll make a certain protein. And then a combination of proteins is then, sorry, I'll start that again. So every combination of nucleotide bases makes up an amino acid, and then that combination of amino acids then makes up a certain protein. So for instance, TGG makes tryptophan, which is a precursor to serotonin. Okay, so the first stage in gene expression is gene transcription. So what happens here is DNA is turned into an RNA strand. So this originally starts by transcription factors attaching themselves to the promoter region of a DNA molecule, which would be right here. And then that DNA molecule is then unzipped by a protein called RNA polymerase. And then once that DNA molecule is transcribed into a messenger RNA, it goes to the next step, which is translation, which is the second step. And what happens here is that the messenger RNA strand leaves the nucleus and then attaches itself to a ribosome, which are found on the endoplasmic reticulum. And then when they're attached to the ribosome, what happens is transfer RNAs also attach to the ribosome, and then here, I'll actually draw that out. So when the messenger RNA is attached to the ribosome, there's a bunch of transfer RNAs that are floating around within the cytoplasm, and then they also attach themselves to the ribosome. And then at one end of the transfer RNA, you have what is called an anticodon, which is a match to the codon here. So for this instance, it would be GCC. And then this is the transfer RNA here, tRNA. And then at the other end, you have that amino acid. And then this slides along the ribosome, starting with CGG and then UUU, and then GCC all the way through that entire gene sequence. And then at the end of it, there's a terminal sequence, which pretty much just ends gene translation, and then the amino acid chain is produced. So kind of to recap that, so the process from DNA to mRNA is transcription, and then from mRNA to an amino acid chain is called translation. So an amino acid chain is, an amino acid is typically, they all have the same structure, which is a central carbon atom, a hydrogen atom, an amino group, and a carboxyl group. And the only thing that distinguishes between all 20 types of amino acids is a side chain. So an amino acid forms, or a combination of amino acids together form a polypeptide chain. And then that polypeptide chain then folds into a specific configuration, which makes up the actual protein. So for instance, an enzyme, a neurotransmitter, or a neurotransmitter receptor. And once that protein is finished, once it's folded, it moves to the Golgi body where it is packaged in a lipid or sugar vessel, and then it is transported down the microtubules to the terminal buttons at the end of the axon where it waits to be used. So any questions on that? I know I kind of went through that fast and it seemed kind of complicated. Yep.
So neurotransmitters would be like an example of these proteins that are synthesized?
Yeah. So it's not only neurotransmitters. It would be pretty much any protein that the cell needs.
What, for neurons specifically?
For neurons, it would be neurotransmitters. It would be the receptors for the neurotransmitters. It would be growth factors and the neuropeptides.
Neurons use a lot of proteins together, like all the microtubules, those are all proteins. Every part of the neuron is made up of proteins. So it's all those, wear and tear things they need to maintain, et cetera, and depending on the activity of the neuron as well, whether or not it's being used or not, and what it will create.
Is everyone good on the gene expression stuff? So basically all you would probably need to know for the midterm is DNA to mRNA is done through transcription, and the proteins or enzymes that help there would be RNA polymerase. And then mRNA to an amino acid chain is done through translation, which involves the ribosome and the transfer RNA. And that's pretty much it. Okay.
Any other questions about that? Oh, yeah.
Just regarding the diagrams, like the diagrams or just what you just said?
These diagrams, no.
Anything else? Okay, so now we're going to move on and talk a little bit more about stuff that you guys have already learned. So it'll be more of a review for everyone. We're going to talk about the action potential specifically. And I don't know how to use this stuff, so I'm just going to use the mouse to kind of dictate where I am. So as we talked about, everything that went on with the transcription, translation, occurs in the nucleus, in the soma, so the neuron cell body, and then the microtubules transport all that down to the terminal buttons at the end of the axon, where it then forms a synapse and can communicate with other neurons, back to itself, etc. So basically what we're interested in is how these signals get relayed to these other neurons, and that is through these action potentials. Oop. How do you get it to go forward?
I just use like the arrow.
Ah, all right. Oh, I messed it up now. Sorry, guys. There we go. Nope. There we go. Okay. So the first thing that we need to talk about is your resting membrane potential. And that is the difference between the voltage or the polarity between the extracellular side and the intracellular side of a neuron or parts of a neuron. Okay? So if you look at, there's different things that make up and determine this polarity. So if we look at the ion distribution, that's one thing that affects its polarity. So inside in the intracellular area, so inside the axon, so this is the extracellular, intracellular. So inside the axon, there's a higher concentration of protein ions, which are negatively charged, and you have a higher concentration of potassium ions, which are positively charged, compared to extracellularly. So you can see there are more of these two compounds, proteins, ions, inside the axon than there is outside at resting potential. The opposite is true for sodium ions as well as chlorine ions. There are higher concentrations of both of these ions on the extracellular side compared to the intracellular side. Okay, so this is what occurs during resting membrane potential and what can contribute to that difference in charge, right? Other thing that is important is like within the membrane, there's these pores and channels that are also made up of proteins, and they actually contribute to maintaining this resting membrane potential. Okay, so there's certain ions pass through certain pores. So here is your potassium channel, where potassium can freely move in and out. Although at resting membrane potential, this does not occur because of electrostatic forces, right? And also through the concentration gradient, right? So potassium wants to move out because there's lots of it in here and not very much out there. But the outside is more positive than the inside. So it also wants to stay in to equalize that difference between it, right? So there's that pressure for it to go out against the, go with the concentration gradient, but there's that pressure for it to remain inside to increase the positiveness, if you will, the inside of the neuron. Sodium, on the other hand, is not open at resting membrane potential, so it's not free to move in and out. And sodium then, therefore, can only remain on the outside of the axon and the extracellular space and can't freely move in and out to the intracellular space, also contributing to the high concentration of potassium ions within the intracellular area. Okay? There's also this thing called the sodium-potassium pump, which also helps to maintain resting membrane potential. For this to happen, 3 sodium ions get pumped out of the axon. So this is the intracellular space here and the extracellular space here. So 3 sodium ions, because there is some sodium leakage into the axon. It's not completely barren of these sodium ions. And so when these sodium ions get leaked into, or that are already present there, they are pumped out for every 2 potassium ions that are pumped in to help maintain that, that difference in polarity between the extracellular side and the intracellular side. So that difference, does anyone know what the resting, on average, the resting membrane potential is? There is some discrepancies between the way it's reported. Negative 70 millivolts, that's right. Okay, so it doesn't mean that the inside is negative and the outside is positive. It just means relative to the outside, the inside is more negative. So I know that's something that people sometimes find a little bit complicated because of this. There's so many potassium ions inside, how can it be negative? But it's relative to the outside that it makes it negative. Does that make sense? Any questions about that? Okay. So again, this is kind of just what I said written down. Resting membrane potential, again, is maintained through these diffusion along the concentration gradient, so that leakage or transport of these ions in or out of the extracellular space. Also through active transport, so that's the sodium-potassium pump. They're actively using energy of the cell to pump these sodium ions out and pump these potassium ions in, as well as electrostatic pressure, so that's your negative positive charges. There's that pressure for potassium to be on the inside of the neuron relative to the outside of the neuron. And also permeability at the ion channel. So I was talking a little bit about leakage for the sodium ions. Okay. So something else that's also really important is postsynaptic potentials. Can anyone explain to me what a postsynaptic potential is? Anyone been studying so far? Probably not. Oh, yes, yeah.
When a nerve, an axon terminal fires into another cell, basically.
Right. So it's a neuron is receiving messages from another neuron through the influx of ions, and this changes the voltage of the membrane around that area. Okay. So you can have inhibitory and excitatory postsynaptic potentials. So for inhibitory postsynaptic potentials, they create a hyperpolarization. Okay, so they actually decrease the polarity of the membrane to about minus 73 millivolts. And this is done through an efflux, so an exiting of potassium ions from inside the intracellular fluid, and a influx of chlorine ions. So it makes the inside of the axon or the cell body more negative. The opposite happens through excitatory postsynaptic potentials. It leads to a depolarization of that region, which may or may not lead to an action potential. So remember that these postsynaptic potentials are graded, and they add up over time to determine whether or not an action potential would be produced. You have to reach a certain threshold in order for one to be produced, and if you reach that threshold, then one will always be produced at the same rate. But if it's not reached, then essentially nothing will be done. There will be no action potential that be contributed, that will be triggered. Okay, so for depolarization, you need a rapid influx of these sodium ions through the sodium ion channels into the inside of the neurons, so inside the cell membrane, the axon, et cetera, depending on where that synapse occurs. Okay. So this is what I was talking about, how they were graded and they were summed up together. So you can have temporal and spatial summation of all these postsynaptic potentials. So they'll be occurring all over the cell body, the dendrites. They can even occur on the axons, and they will all determine whether or not the threshold will be reached to allow a action potential to be generated. And what happens is all of these come together at the axon helix, and if the threshold is reached here, an action potential will be generated. Okay, so what's important here is your temporal and spatial summation. So postsynaptic potentials that are closer together in time will lead to a bigger depolarization effect. Closer together on the neuron will also lead to a bigger depolarization effect. Whereas, say you just had an excitatory postsynaptic potential here and here, those are pretty far away on the neuron, and they won't add together, I guess, and have a synergistic effect and lead to an amplification of these graded potentials within the cell soma and then contribute to generating an action potential. So think about this as if you have like a bucket and there's like a hole in it and you're pouring water into it. And if you think of pouring water into it as being excitatory postsynaptic potential and in the hole, water leaving that as being inhibitory postsynaptic potentials, at some point, if you pour enough water in it, it will overflow. You'll reach that threshold for water to leak over the edge, just like you'll reach that threshold for an action potential to be generated. If you are pouring water in too slowly, it will leak out all the time, and you will never be able to generate an action potential. Does that make sense? Okay. And I think I might have this on the next slide. Oh, yes. So action potentials are an all and nothing or a nothing loss. So they either happen or they don't. Okay, so there's nothing, there's no such thing, and this is a common thing that we do see on written on exams, that there's weak action potentials and stronger action potentials, and sometimes different scenarios will lead to an even bigger action potential, which is completely untrue. They either happen or they don't, and they're all the same grade. What is different is the postsynaptic potentials. Those can be bigger or smaller depending on what is going on around the neuron and the input it's receiving from other neurons. Okay, so action potential is a large temporary reversal of the polarity. So if you remember, where is it? Here, this is at resting potential. And what happens with an action potential is the inside becomes positive and the outside becomes more negative relative to each other. So that's the reversal of polarity that I'm talking about, the reversal of the positive and negative charges. And they are relatively long compared to excitatory postsynaptic potentials. Okay, so they last about approximately 1 millisecond, which is relatively short, thinking about it that way, but excitatory postsynaptic potentials and inhibitory postsynaptic potentials are much quicker in comparison. Okay, so now if you want to think about what's actually going on at the ion level while an action potential is occurring. So you have to have an influx or a depolarization as a result of an influx of sodium ions large enough to be able to reach this threshold and generate an action potential. Once that happens, so your sodium ions are going in. Once that happens, you have this big surge and the reversal of polarity that travels along the axon over and over and over again, where you have influx of sodium ions, and eventually the potassium channel is also open. This occurs a little bit more slowly. Potassium ions also open and cause an efflux of potassium outside of the neuron. This is done in response to the increase of the positive charge inside the neuron. If you remember, potassium ions are positively charged, and so to go with the electrostatic pressure, they now need to be released from the inside of the axon or the inside of the membrane to the extracellular side to return to resting membrane potential. So it's called repolarization. And then the other thing to point out too is that as a result of this large influx of sodium ions, at some point the non-voltage gate on the sodium channel actually closes to prevent any more influx of sodium into the intracellular part of the neuron, to allow, again, the intercellular spark to become more negative again. Okay, so it's the combination of the closing of this gate 2, the non-voltage based or sensitive gate on the sodium channel, as well as the efflux of potassium ions that allows the membrane to return to resting membrane potential. Another thing, so this part here, the absolute refractory period, which is what this part is referring to, is a point in time where another action potential cannot be generated. And that's largely due because of this closing of the non-voltage sensitive gate on the sodium channel. If you remember, the depolarization from the action potential is a direct result of this influx of sodium ions. So if that is blocked, you can't generate an action potential. The other point out to make is this relative refractory period. So when the sodium ion channel still remains closed, the neuron is still pumping out potassium ions. And so it kind of overshoots the resting membrane potential, and that's why you have that little dip that goes down to minus 73 millivolts before it returns up to minus 70 millivolts. Because the neuron is still pumping out potassium, and it takes a while for it to be able to return to its minus 70 resting potential as well. So anyone know how it returns from this hyperpolarized state to its resting membrane? What factor is important in returning that? Yeah. The pump, right. It's the sodium-potassium pump. So it's pumping out 3 sodium ions, and for every 2 potassium ions that are coming in. And that's what helps it return to the resting membrane potential. Something else to point about this relative refractory period is you can actually generate an action potential here. So if you look here, the gate 2, the non-voltage sensitive gate, is actually open at this stage. So it means an influx of sodium is possible, and thus a depolarization or an action potential is also possible. Although, since resting potential is minus 70, and right here it's minus 73, you're going to need an even more, larger response from your excitatory postsynaptic potentials in order to generate it. Because now you're not going from minus 70 to depolarization, you're going from minus 73 to depolarization. Does that make sense? Yes?
On the slide that you had for resting membrane potential, you showed the potassium pumps as being a channel. So on the axon, they're a gate. On the postsynaptic button, they're a channel?
No, they're all gated. It's not a great picture. I think that's one from your book, if I remember correctly. It's not the greatest picture, but for what the no, there is a gated, because it can open and close, but it's not. The thing is, when you think about ion channels, they don't actually have these little flap that goes up and down. It's just a way of kind of demonstrating it. It closes in different ways depending on different scenarios, things binding to it, etc. So basically it just means that nothing can pass through it. And so this gate part, like we talk about it as being a gate, but because it's easier for us to think about it in that sense, but it's not really like an actual like flap that opens and closes.
So it's not a difference. Like, in the book, it does make a differentiation between something that is a channel and something that is a gate. In both cases, it's actually a gate for this particular protein.
In both cases, they can be open and they can be closed.
Okay.
So yes, you are correct when you are talking about there is a distinguish between things that are gated and things that are channels, but they both can be opened and they both can be closed. Yeah.
Is the difference that a channel just opens more often than a gate?
Well, the difference is depending on whether or not it's voltage gated, right? If it's voltage gated, then it's sensitive to changes in polarity, so changes in whether or not things are more positive or more negative. So it will open in a more negative environment or it'll open in a more positive environment, whereas a channel will more likely open in response to something maybe like a receptor. Something binds to a receptor. It can, like your ionotropic receptor. Something binds to it, it opens the channels, it allows ions to go through. When it's gated, again, sometimes it depends on whether or not there's changes in polarity. Does that answer your question? Okay. Any other questions? Okay. Okay, so how do you get this to click on it? Do you like exit? Sorry, I'm not really good with, ooh. So this is a little animation that I found. Can you help me? Thank you. So it's a little animation that I found. The first part of it, I can't figure out how to skip the beginning part of it without skipping halfway through all the other part, but it basically just goes over exactly what I talked about. And then there's like these little exercises embedded in it where you guys can tell me what I'm supposed to do. And it's well done. If we can't get it to go, then we can try it out later on. All right, well, we'll try it after again if we can't get it to work during the question period. All right. So the other thing I wanted to talk about too was some axons are different from other axons. So some of them contain myelin sheath that are coating the axon and some are not. Does anyone tell me the difference between axons that are myelinated versus axons that are unmyelinated? Yeah.
As long as they're myelinated, the signal travels faster throughout it.
That's exactly right. Right. So what she said was axons that are myelinated compared to axons that are unmyelinated, the current or the action potential actually travels quicker or faster down the axon than ones that are unmyelinated. Okay, so this is an example of an unmyelinated axon here. And when an action potential is generated, you see it starts out with your negative intracellular space and your positive extracellular space. When an action potential is generated and you have an influx of sodium into the ion, it actually reverses this polarity. And this is what travels down the ion. It's the reversal of polarity. And once the action potential is finished, so it's in repolarizing or returning to resting membrane potential, it reverts back to its original polarity. So you can see here that this is where the action potential is occurring, and you have that reversal of polarity. And it returns to membrane potential as you travel down, and this is where the polarity is reversing. And then it returns to membrane potential, and then this is where, again, the polarity is occurring, and you can see that's where the action potential is also occurring. So it just travels down. So it's kind of a repeated kind of an assembly line of steps that occur along the axon. When axons are myelinated, and just to point out, so the glial cell that's important in myelination is your oligodendrocytes. this is important for neurons that are in your CNS. For your nerve cells that are found in the peripheral nervous system, they're actually myelinated by Schwann cells. In the CNS, just an interesting fact, myelination, like once it's been destroyed, these cells can't remyelinate. Whereas Schwann cells in the PNS, myelination can actually reoccur. So that's why diseases and and you probably will talk about this a little bit later on, but diseases like multiple sclerosis don't have a cure because we can't remyelinate these neurons that have been lost. Whereas anything that occurs in the peripheral nervous system, Schwann cells can actually regenerate. So the purpose of it, as we mentioned before, is to speed up the neural impulses in the axons. And you have these pockets where it's myelinated, and then an area where it's unmyelinated, and it kind of alternates back and forth between these two things along the axon. And the unmyelinated areas are called nodes of Ranvier, and they are not myelinated, but they have all your voltage sensitive channels, so your ion channels that are responsible and important in generating action potentials. Okay, so another word for an action potential along a myelinated axon is called saltatory conduction. So saltare meaning to jump or to hop. So the action potential actually jumps from node to node to node in kind of loosely speaking. It doesn't actually jump over. It kind of, you have your action potential here. The current passes through here. It doesn't need any axon or ion channels to open or close in order for that to happen. And then it reaches another node of Ranvier where the ion channels need to open and close in order to generate that action potential, and then the current again is passed through the next myelinated area. Okay, does that make sense? Any questions? Yes?
I've always wondered, is it just because the myelin sheath is just more conductive, or does it just block the channels so it doesn't, so the channels don't actually activate? It's just kind of a curious question.
Well, I think, and I just read this, and I can't even remember the answer now, so I'll have to look it up again, and I can respond on the discussion board. But I think it's the fact that it doesn't contain these voltage-sensitive gates, so you don't have that extra time waiting for these to open and close in order for it to continue down, so it can just pass through that area. And yes, in a sense, the myelin sheath provides a protective, like a, I guess, coating, just like when you, excuse me for butchering this term, but when you have wires for like construction stuff and they're coated and everything to kind of increase the speed of the transmission. I don't know what any of these actual words are, so sorry if it doesn't make sense. Those when they're coated, the signal gets transmitted faster. So it's kind of the same thing. It's coated, so then it's passing through the signal a little bit faster. Does that make sense? Okay. Okay, so I have another thing where we can look at that after again. All right. So once the action potential reaches the terminal button, so that's the end of the axon, so that's this little kind of, I don't even know how you would describe that shape. Basically, it kind of kickstarts a few things that occur in this area that's specific to this area. So these are microtubules that Rob was talking about earlier, and they're transporting down these proteins. And in this case, they're transporting down these vesicles, which later occurs in this area, is they're packaged with these neurotransmitters that have also been synthesized in the neuron and transported down these microtubules. What happens is they're packaged into here. They're also, when a neuron is not activated, they are packaged into these other granule cells here until they are needed for use. When an action potential is delivered, it causes these synaptic vesicles to actually embed in the membrane. And through exocytosis, it actually releases its contents into the synapse. So that's that little gap in between the terminal button on this axon and whatever it is connecting to. So it can be another, a dendrite, an axon, a soma, etc. So wherever the synapse is formed within the CNS, it releases its contents where it can then, these neurotransmitters can then bind to receptors found on the postsynaptic neuron and either cause an inhibitory or an excitatory postsynaptic potential to occur. So it depends on whatever neuron is delivering the message as to what message actually gets delivered, whether or not it's an excitatory or an inhibitory message. Okay, so basically these receptors bind to other receptors on this area and they open ion channels and cause the influx of sodium ions in the case of an excitatory postsynaptic potential, or cause an efflux of potassium and an influx of chlorine ions in the case of hyperpolarization or an inhibitory postsynaptic potential. The other thing to point out too is in this diagram, it's calcium that's important in the release of these neurotransmitters to the synaptic cleft here. Okay, so you generate an action potential. This influx of calcium ions bind to these synaptic vesicles and cause them to merge through exocytosis to the cell, the end of the terminal button, and release its contents. Does that make sense? Okay, so the last thing I'm going to talk about are the different receptor types. Yes? The extracellular side. There's lots of ions floating around in your extracellular space, and that's where they all come from. Did I see someone's hand on the side? No? Okay. So we have two types of receptors, your ionotropic receptors and your metabotropic receptors. So for the ionotropic receptors, the neurotransmitter actually binds to this receptor, causing the pore to open or close depending on what the neurotransmitter is released. So in this case, it is binding to the receptor and causing it to open, allowing the influx of ions. So this is much quicker than your metabotropic, as you'll see in a second, and as you probably remember, because it causes an immediate change and reaction, allowing the influx of these ions into the postsynaptic neuron. Metabotropic has an extra step for opening these ion channels. So it binds to the receptor, and as Rob mentioned earlier, it requires the alpha subunit that's bound to this G protein or receptor that is embedded in the membrane to be released. And then you have to wait for this alpha subunit to bind to its appropriate ion channel in order for it to open and allowing the influx or the efflux of these ions. Okay? Another way is an even more slow way, and I think this is the way that Rob was talking about before, is you have your neurotransmitter bind to your G protein, causing a change in conformation and allowing this alpha subunit to become released, where it then binds to an enzyme, which can then go and change and cause all these downward cascade of changes within the neuron, which can also affect gene transcription and all these other proteins that are created within the neuron that will effectively alter what is going on in the neuron. Okay, so all of these things are connected. So all these action potentials are connected to what Rob was talking about before, the internal workings of the actual neuron, what's going on, the basics of what cells do. So translation and transcription. Does that make sense? Yes?
So it can also create new channels, right?
Yep. And that's based on what's going on in the second messenger in this one here, because it's new channels are generated, and the upregulation or the downregulation of these channels are turned on through gene expression.
So is there like a limitation on each neuron on how many channels there can be? Or
No.
does it ever like stop making new channels?
It can. So, but that is in response to environmental changes, right? So say you are using one neuron to another neuron, that connection keeps, being used over and over and over and over again. And at some point, the neurotransmitter that's being released from the first neuron becomes, depleted. It's not being made as quickly. Then in response, the second neuron may develop or may create more receptors in order to pick up that neurotransmitter more effectively. The other thing is the opposite. If too much of a neurotransmitter is being released, it may minimize the number of receptors that are present on its postsynaptic side, so it's not being constantly bombarded by action potential. So there is kind of a balance that does occur. And so the thing about it in this way is like drug use, which you will talk about with Kim, is drug use can affect the amount of neurotransmitters that are released that are kind of floating around in your synapse. And so that will definitely affect the number of receptors that are on that postsynaptic side. Any other questions? All right. Do you want me to see if I can get those things working, or do you have any other questions about any other of the course material? So we have lots of stuff we can potentially talk about. Yes?
During an axo extracellular synapse, what would actually cause the synapse to just dump everything into the extracellular fluid as opposed to having to bind to another part of the neuron?
Like, sorry, can you say that again?
For an axo extracellular synapse, so when the axon just dumps whatever's in the synapse into the extracellular fluid, why would that occur instead of the axon finding another part of another neuron to synapse onto?
Well, it all depends on like a bunch of scenarios that occurred to form that synapse in the first place. Because when you look at an axon and it has like one little button like that, it's not really necessarily the case. It can have multiple offshoots of things, and so maybe in response to being activated too much, some of these or not being activated enough, and you're pruning out these inactive synapses, that it causes it to synapse somewhere else, and where it doesn't want to synapse on another neuron to propagate the signal. So there's a lot of different scenarios that can be in place, and I'm sure that we don't fully understand why those things occur, because there's lots of things about neuroscience that are basic that we don't know why that even happens, but they do. We just know that. So that sort of half answers that. Yes?
There was mention of type 1 and type 2 synapses, type 1 being excitatory and type 2 being inhibitory, and that type 1 synapses seem to always occur in the dendritic tree, so axodendritic, and axosomatic are always type 2. Was I misinterpreting that from the textbook?
No, that sounds about right. And if you think about it,
so any axosomatic synapse is going to be inhibitory?
not necessarily.
Okay.
There's plenty that are excitatory as well, but the point I think the book is probably trying to make is that they tend to be inhibitory more. Any other questions?
Can you just repeat that? It was type 1 is excitatory.
Right. Type 1 synapses are excitatory, and type 2 are inhibitory. Can you make it work? Oh, okay. So I just thought I'd play a few animations to kind of show a different way of how these occur. So this is neurotransmission. And I think it actually explains it. Is there a volume button?
I hope so.
So it's just another way of looking at it.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule.
So let's just use an example of acetylcholine.
Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive.
So what kind
If
of
the membrane
receptor is that
potential
one?
reaches threshold level, an action potential
Example of
will be
ionotropic
produced.
or metabotropic? Ionotropic, right. Okay, and then this is the other one that I wanted to show, and you can just click on that like blue button. So this is an unmyelinated axon on the top. So it shows when an action potential is being delivered, the reversal in polarity as it travels along the axon, and its return to its resting membrane potential once it's passed. And then if you look at the way a myelinated axon occurs, you see that the current is passed through the myelinated area, and the ion channels are present on the unmyelinated areas, speeding up the transmission. Okay. Make sense? Okay. Any other questions, guys? But it doesn't have to necessarily be about any of this stuff. It can be about other parts of the course as well. I'll post this link on the class discussion board, so if you want to look at it later, you can.
For the short answer questions, are we allowed to do point form?
Oh yeah, actually, yeah, we really like point form because it's a lot easier to see where you are when you're. Sometimes people use like big introductory sentences. We don't like. It's not going to give you any extra marks to have any of that stuff there. So if you just want to be straight to the point and put it down there, that's fine. Yeah.
On the diagram questions, are we going to have to label stuff like dorsal, ventral, medial, or is it really just going to be parts of the brain?
It could be anything, any kind of diagram, and yes, dorsal, ventral, all that can be depicted in a diagram, right? So yes, that's fair game for the class. Yep.
Will it be single word labeling, or will it be like description labeling?
Oh, it'll be like, yeah. Or I mean, like if it's like a term, then it might have 2 words, but it will just be like, yeah, just a kind of a description. No description, sorry. Anything else?
I guess the last thing we're going to mention is we are having like virtual TA hours tomorrow from 7 till 10. So you guys could log on that. I think it's called the Big Blue Button.
It's
So it'll be
in
on
your
there.
C, if you want, I think you can access it through C Learn, and kind of on the side where you have, class discussion, TA discussion board, it's at the very bottom. And I can't remember what it's called, but something along online TA hours. And it has just kind of like a little blue button, and there's a B in it. And it's what the WebCT chat used to be. It's a little bit better, I think, now. But you can just log on through there. And I think you might need to be on Internet Explorer for it to work. If you're using Firefox or something else, it might be a little buggy, because I know that's what the old program used to be like.
Well, I think we're done. You guys could also ask us questions after class if you want.
We'll be around for a bit. Or email us. All right.